Hello everyone. Let's take today's topic. Today we'll discuss about stomata and transpiration. I gave you the various concepts of transpiration earlier. We'll focus on the stomatal transpiration. Before starting of this chapter, this topic of this chapter, we'll have to understand about the structure of stomata. And then after we'll focus on the role of the stomata in transpiration. I taught you that above 90% water is lost through stomata. We'll take structure of stomata. It is a quite simple structure, you need to understand only and try to focus on the concept while in understanding structure of stomata. What we are getting here, we are discussing about the structure of stomata, and then we'll understand the role of stomata. Here we should understand this one is thicker than inside there is, I taught you earlier, it is a cell, so we have a nucleus also, chloroplast also. We should only that draw this one, there is no need to write anything. These two structures are known as gourd cell. We are calling gourd cells. When we see the gourd cell, what we are getting, this is the outer membrane of gourd cell, outer membrane, and this one is known as inner membrane. What we call inner membrane. Outer membrane, when we see, we can see it is thin in nature, thin, and inner membrane, it is thick. Always focus on these 2 points. These are very important points. We are understanding about the structure of stomata. This structure we call gourd cells, and it is having 2 membranes. First one is outer membrane, it is thin in nature, and inner membrane, that is thick in nature. And what we are observing here, try to get, we are observing a pore, and that pore we call stoma. And we can also say stomatal aperture. All these words are related to an opening. There is an opening, and that we have a structure that is known as stoma. This structure is, we are calling open stomata. And there is another structure also. Try to get this one, open stomata, and when we draw another, this stoma is not there. Or we can say that it is not as much as widen. You can see the difference, not as much as widen here. This same structures are there, only you need to understand there is a one concept. There is also a thick wall, this is also a thick wall. This is known as closed stomata. What we call closed stomata. Whenever we have a closed stomata, there is no transpiration, or least transpiration we can say, because the other ways of transpiration are also there, but 90% of water is lost through stomata, and when stomata is closed, there is no chance of the transpiration. That is the logic we need to apply only. we can see the difference between two structures clearly. Now we'll put these concepts what we discussed in transpiration, when we have opening of stomata and when we have closing of stomata. There is no doubt in role of transpiration of stomata. We understood at the daytime, whenever we have a sunrise, we have the starting of stomata, we have the starting of transpiration, sorry, we have opening of stomata, and whenever we have the opening of stomata, that pore is appeared, we will have a transpiration. And at night, when we have a sunset, we will have a closing of stomata, and that pore is lost, we have no transpiration also. That is the concept. Now our understanding will be based on when stomata opens and when stomata closes. That is the concept. We'll focus on the mechanism of opening and closing of stomata. Try to understand what we are getting here. Mechanism of opening and closing of stomata. This is the topic we are going to discuss. Mechanism of opening and closing. When we have this one mechanism, we need to only imbibe 2 points in our mind. We discussed earlier turgidity and flaccidity. What is turgidity? Whenever we have entry of water by the process of endosmosis, it creates a pressure, and that pressure will result into the expansion of a membrane, and that cell swells. That is turgidity. Flaccidity, when water is moving out from the cell, that pressure is released and we have the shrinkage. That condition is known as flaccidity. Already we discussed in previous chapter. I explained you only to recall all these points. Let's take the mechanism. We have to focus only 2 points. When guard cells become turgid, stomata gets open. That is the concept. Easy to understand. This is the guard cell. Whenever we have entry of water, try to understand in this diagram, because we are talking about that one, opening of stomata. This is the guard cell. Whenever we have entry of water, suppose we have entry of water, water enters, and after a certain extent, that water creates pressure on this wall, this membrane. This membrane is flexible, so it starts shifting. So when it shifts, it will also move, and when it will move, we have the appearance of stoma, and that is the opening. Very easy to understand. Whenever we have a pressure and when the membrane is flexible, obviously it will be shifted, and when it shifts, we will have the shifting, because this membrane is also the part of this one. Whenever it is moving, this will also move, and when it moves, we have the opening of the stoma. That is the first point. What is the second? When guard cells become flaccid, stomata gets closed. When guard cells become flaccid, it means we have the shrinking. Try to get this concept in this diagram. Here we have the filling of water, so that we observe the expansion. Whenever we have the exosmosis, water is moved out. So when water is moving out, these cells they start shrinking. When they shrink, we will have that was shifting of this membrane also, and whenever we have the shifting of this membrane, we will have closing of stomata. That is the concept. Easy to get it. It means opening and closing of stomata depends upon turgidity and flaccidity. That is the core of this topic. Now you have to understand about how we have change in turgidity and flaccidity. That is the matter of discussion. We need to go through what are the causes which are making change in turgidity and flaccidity and how it is happening. We'll take in next video.